Okay, so it's a pleasure to introduce Frantz. Frantz also is originally from Gabon, from near Equatorial Africa, and that's where he got training in geology, and after it he went to France. He did his master and PhD in University of Poitiers, mainly focusing on clay mineralogy. It's one of the world centers still working on clay mineralogy and implication for environment and genetic conditions. During his PhD, he and his PhD advisor Abder El Albani discovered fossils in early Paleoproterozoic Frantz-Gerald succession. And after his PhD, he moved for postdoc to South Africa, to University of Johannesburg, where he worked with Axel Hoffman and other people. And at that time, he continued to work on Frantz-Gerald succession, but also branched out to work on Mesoproterozoic succession in South Africa on Angola supergroup. And I guess it would be part of his topic that he will discuss today. And after postdoc at South Africa, he wanted to improve his analytical skills. So he joined University of Tuebingen, Ronnie Schoenberg, and worked on various non-traditional stable isotopes. I'm aware he worked on zinc, selenium, iron, few other isotopes, and related to both Frantz-Gerald and Pangola supergroup. And so it's a pleasure to introduce Frantz. Currently Frantz looking for permanent or temporary positions. So if you any senior people here looking for postdoc or temporary instructor or has permanent position open, feel free to contact Frantz and to encourage him to apply. So with this, I pass it to Frantz. Frantz, can you unmute yourself and make it?
Share your screen, Fritz.
Yeah.
Okay. All right, we have the, oh, that's right, you have to do the swap.
Is it fine?
That's good, great, yep.
Okay, thank you. Thanks for the introduction, Andre, and thanks for having me here, and hi to everyone. Today I'm going to show you some mechanism of manganese precipitation in the Mesoarchean Mozambique group of South Africa. And it's a collaborative work with various people that you can see down there. And just to give you some feedback, just going straight to the point, the first permanent raise of atmospheric oxygen is widely accepted to have occurred sometime earlier during the Paleoproterozoic, and the Archean is widely described also to have remain permanently anoxic, but with some episodic development of localized atmosphere and ocean oxygenation. Some biogeochemical proxies, modeling, and molecular clock estimate suggest that oxygenic photosynthesis will have started as early as during the Paleo-Archean. And what I will show you today is what is happening in the Mozambique group, because one of these extensively described surface ocean oxygenation is host in the Mozambique group sediment. So why the Mozambique group? Because it's characterized by manganese-rich banded iron formation and some ferruginous shells. Manganese is host in early diagenetic carbonate minerals, and the precipitation of this manganese is described to occur as oxide from seawater. And it was mainly the capacity of manganese to remediate some other metals that was used in a study published by Planaski et al. In 2014 that was showing that this positive correlation between manganese enrichment and light molybdenum isotope in banded iron formation of the Mozambique group. And then this is particularly because manganese oxide preferentially absorb the light molybdenum isotope. So it was quite a strong evidence that manganese oxide was formed in the Mozambique group basin. And once it was already proven that manganese oxide was there, the next step was to assess the main mechanism that would have been responsible for this manganese oxidation. And three main possibilities can explain this oxidation of manganese in the Mozambique group. So the first pathway will be oxidation of manganese under oxygenated condition, which require oxygenic photosynthesis. The second possibility is by photochemical oxidation under anoxic condition. And the third one will be through an oxygenic photosynthesis. But an experiment by Anbar and Hollande in the early 90s shown that the photochemical oxidation of manganese will be strongly inhibited in seawater with dissolved iron. So this possibility was ruled out. And by the time this article was published, there was no example of an oxygenic photosynthesis oxidizing manganese. So the most plausible explanation at that time was only the oxidation of manganese under oxygenated condition. But more recently, an experiment made under sulfidic system shown that manganese can be oxidized through an oxygenic photosynthesis, and then ruling out any possibility for using the oxidation of manganese as compelling evidence for oxygenated condition in deep time. And what I will show you today is how the sequestration of manganese in the Mesoarchean Mozambique group can only be explained by oxygenated conditions. So let's just go back to the Mozambique group and some geological information. It's a Mesoarchean sedimentary succession of the Pongola supergroup, which is deposited in the eastern part of the Cap Val Craton in South Africa. So although the sedimentary succession is characterized by 11 lithostratigraphic formation, so far we only studied the lower part, which is characterized by the Sinkeni formation, the Tombe formation, up to the Talu formation. So some sedimentological analysis show like basically shallow water deposit. As you can see, some interference ripples, symmetrical wave ripples, and mud cracking showing like tide influence shallow marine environment with episodic exposure. And then another part of the section is characterized by some mid-depth deposit where you can see some wave ripples with some reactivation surface and some graded bedding with some rhythmite and then Umuki cross stratification indicating deposition between fair weather and some wave phase at the transition between the inner and outer shelf, and also some deep water deposit where banded iron formation precipitated, characterized mainly by planar laminated sedimentary structure away from any wave influence. So together with multiple drill core logging, we describe depositional setting from shallow water in the soft west part of the basin, the white follows it here, to deep water toward the Nongoma area, northeast from the basin. And then we evaluated the variation of manganese concentration in this various deposit, and it's clearly appear that we have more higher manganese concentration in shallow water, and then this manganese concentration decrease as the water depth increase. And then we can see that the manganese iron ratio is also increasing considerably as the water depth decrease. So we have more manganese precipitation in shallow water and more iron precipitation in deep water. And then just to check some petrography to see where manganese and iron are mainly host. And then we see that in shallow ferruginous shells, manganese is mainly host in carbonate, photosynthesis, siderite, cutenorite, and iron is mainly host in carbonate and some phyllosilicate like chlorite. In mid-depth, associated with banded iron formation, manganese is mainly host in carbonate minerals like siderite, while iron is host in both carbonate and oxide like magnetite. In deep water, so there is basically no manganese, and iron is mainly host in phyllosilicate like strychnomelan and chlorite. So we remeasured molybdenum and iron isotope, and as we can clearly show that in shallow water with higher manganese concentration is also there, that we have light molybdenum isotope composition and light iron isotope composition. As the water depth increase, we tend to have unfractionated molybdenum and iron typical from the hydrothermal signature. So according to these various biogeochemical proxies and petrographic and sedimentological analysis, we found progressive oxidation of manganese as the water depth decrease, and where deep water basically upwelled across the redox line, and manganese was mainly oxidized in shallow water where more oxygen was produced, and an additional pathway of iron oxidation was driven by manganese oxide in shallow marine water. So, and then molybdenum, light molybdenum isotope was mainly absorbed in manganese oxide, which gives the isotopic trend that we saw earlier from shallow to deep water. So to try to constrain the mechanism of carbonate formation, we measure the carbon isotope And then we found that they were highly depleted compared to Archean seawater here. And then it's clearly shown that the precipitation of carbonate required more depleted carbon source, which is likely organic carbon. So this is supportive that the precipitation of carbonate occur through the remineralization of organic carbon. And this model, which is well known from the literature, show that at least 50% of remineralized organic carbon contributed to the precipitation of this carbonate. But because we sometimes have some band of carbonate layer, like you can see here, one can suggest that maybe carbonate precipitated from seawater before settling to the sediment, but the occurrence of carbonate concretions here in shallow water shells and in banded iron formation clearly show that the precipitation of carbonate occurred during diagenesis, and which is also implying that electron acceptors like iron and manganese oxide were present in sediment during the precipitation of this manganese and iron-rich carbonate. So it's clearly shown that we had dissimilatory iron and manganese oxidation in sediment, driving the organic carbon remineralization and precipitation of carbonate in the Mozambique group sediment. Just to further support this possibility of manganese oxide reaching the sediment, I will just like briefly showing this kinetic model of reducing manganese in completely anoxic seawater column. It's a result from an ongoing work led by Chris Renard. And then the model is based on some parameters from experimental and measurement in natural environment. And it show the depth that manganese oxide produced in shallow water will penetrate in completely anoxic water enriched in dissolved iron. And then here it's just the frequency in C here, the frequency of the depth that you will have only about 0.1% of initial manganese left. And then it was shown in this model that you will have at least 1 meter with 90% of 0.3 to 5 or 6 meters of interval here in B. So below this depth, the entire manganese produced in shallow water will completely be reduced. So this means that if deposited below storm wave base, will not be able to precipitate manganese oxide if the seawater is completely oxidized. The only possibility that manganese oxide can reach if deposited in deep water below storm wave base is if the water column was oxidized to allow the manganese oxide to penetrate the water column and settle toward the sediment. So now that we have evidence of manganese oxide precipitation reaching the sediment in the Mozambique group, let's now see how this manganese is sequestrated in sediment from the Sinkeni toward the Ntombe formation. And then we can see some petrographic images here of the banded iron formations showing some ankerite with very low manganese concentration, but also with some mangan-1 siderite with about 8% of manganese content, and a third phase, mainly kutnoorite with about 5% of manganese concentration. In shallow water, we mainly have manganese, mangan-1 siderite, excuse me, with about 18% of manganese content. But as we go in the Ntombe formation, in deep water, we have more diverse manganese minerals, and the mangan-1 siderite has now about 17% of manganese, and kutnoorite with more manganese also concentration at about 14%. But we have a third phase. If you can see, it will be difficult for you, but you can see the contrast of the colors here, which is a phase darker, which is different than the phase that you have here, which is ferro-1 rhodochrosite, but now with about 30% of manganese. In shallower water, here the contrast is more obvious, where you still have this ferro-1 rhodochrosite with about 30% of manganese oxide, manganese concentration, and mangan-1 siderite with about 17% of manganese concentrations. So this is clearly showing that during the synchrony formation at both deep water and shallow water, you accumulated less manganese in sediment compared to the Ntombe formation, where manganese is more sequestrated into the sediment. And the only possibility you can do that is because during the deposition of the synchrony formation, the bottom water was mildly oxygenated. So it's possible that the reduction layer of manganese was close to the sediment water interface. So this means that when you reduce manganese, there is a possibility to further release manganese back to the seawater, and then just resulting in less manganese sequestration in sediment. But this is a typical example of an external manganese cycling, where you have less oxygen concentration at the bottom water. But in the upper part, in the Ntombe formation, it's possible that the seawater was more oxidized. So you form a thin or oxidized layer into the sediment, which push the manganese reduction layer much deeper into the sediment. So wherever you have manganese reduced into the sediment, when this manganese is going back to the water column, it will be reduced, oxidized again, and then be more accumulated into the sediment. So this is a typical case of an internal manganese cycling, where you have more oxygenated condition in the water column. So from the lower to the upper part, you have an evolution from an external manganese cycling to a sort of more internal manganese cycling. And this, you can only do that if you produce more oxygen in the seawater, or if the redox gradient in seawater completely change. But we have more evidence now for oxygenated condition from this new work published in EPSL, where manganese and iron oxide fractionate uranium isotope. And for uranium to be able to interact with manganese and iron oxide, it needs to be dissolved, and a stable uranium dissolved pool require oxic condition in order to remain stable. So this was further evidence that the surface water in the Mozan basin was well oxygenated. So now that we have proof that seawater was oxygenated, oxygenic photosynthesis was active in the Mozan basin, what then delayed the permanent and pervasive oxygenation of the atmosphere and ocean system during the Archean Eon. So if you remember last week talk, this question was also asked, but a little bit differently. But here we also try to investigate the mechanism or process that could have delayed the permanent rise of oxygenation in the atmosphere and ocean system during the Archean. And to do that, we use molybdenum isotope proxies. And this is because the manganese has a higher redox potential than nitrogen species. And if you oxidize manganese, this means that you will also do nitrification if you have ammonium in this environment, and then nitrification will occur in shallow water, but the nitrification will occur at the redox line, and this should be reflected by some positive nitrogen isotope excursion. But we completely saw that the nitrogen isotope composition in both shallow and deep water record pure molybdenum-based diisotrophic, likely under anoxic condition. But what we also saw is very depleted phosphorus concentrate reservoir in shallow water, which tends to be increased as the water depth increase as well. But the possibility of anoxic condition driving molybdenum-based diisotrophy can be ruled out because manganese and iron were oxidized in this setting. So we believe that the increased nitrogen fixation was due to rapid consumption of small nitrate pool in this strongly redox stratified seawater and contributed to the near to zero nitrogen isotope composition that we have. This can be observed in some Mesoproterozoic setting, or the modern Black Sea is also an excellent illustration of this kind of scenario. So this means that oxygen inventory in this oxygenated surface water was not high enough to build a significant nitrate pool that could have left the isotopic fingerprint of an aerobic nitrogen cycle. So we may have had a low rate of biological oxygen production during the Mesoarchean. So just to finish, we saw that we have evidence of progressive oxygenation in the Mozan basin and low concentration of bioavailable phosphorus rather than nitrogen, because nitrogen fixation was clearly active in this setting. So low phosphorus concentration suppressed the growth and the proliferation of oxygenic photosynthesis and may have delayed the permanent release of oxygen during the Archean. But what we can also notice is like neo-Archean oxygen oasis commonly show positive nitrogen isotope excursion. This means that the size of nitrate inventory was likely much higher during the neo-Archean compared to the Mesoarchean. This is also showing that the geographical extent of Mesoarchean oxygen oasis were likely smaller in size compared to their Neo-Archean equivalent. And this is all I wanted to share with you today.
Great, thank you, Frantz. Great talk. With that, we can open the floor for questions. Looks like Seema would like to ask a question. Or maybe not.
I saw someone asking a question.
Okay, I think Don Sumner would like to ask a question. You can go ahead and unmute yourself, Don.
Thanks, Frantz, for a really interesting talk. I'm wondering about, so one of the problems, of course, with Archean geology is the very limited number of stratigraphic examples. And I'm wondering, like how you can take maybe what you see in the Mozan Basin and what do you think might be unique to that particular depositional environment versus representative of basins and coastal areas that might have been, we might not have the sedimentary record for?
This I understand, this is a good question, I understand, but this is also the concept of oxygen oasis. So this may indicate that you only have localized surface ocean oxygenation, because some other sedimentary succession of similar age don't necessarily show this kind of processes. So this might this may means that you don't have development of an oxygen oasis in others environments. Is it what you were asking?
Yeah, I think it was a pretty amorphous question, pretty vague, yeah. But I think that question of how much local oxygen production you have and how wide it's spread it is really gets at how much oxygen is produced to, say, oxidize the whole surface ocean or the atmosphere. I guess sort of what's the local versus global balance of processes.
Yeah, I understand. This is what we're trying to show by using nitrogen isotope. At the moment, it's difficult to really estimate, but if you use nitrogen isotope, I mean, if the size of the nitrate pool is higher, you should see an isotopic fingerprint of aerobic nitrogen cycling, but we don't see this isotopic fingerprint in our setting. So this means that the size of this oxygen oasis was very small in terms of the size of the nitrate pool also. So like what you can see in the Black Sea, so you have surface oxygenated surface water and deep anoxic water. You have a nitrate pool, but this nitrate pool is not high enough to leave an isotopic fingerprint for aerobic nitrogen cycling. And then we try to compare it with what is happening during the Neo-Proterozoic, where you commonly see positive nitrogen isotope composition, and see that the geographical expand of this Mesoarchean oxygen oasis was likely smaller than the example that you can see in the Neo-Archean.
Yeah, thank you very much.
Okay, thank you. I think Seema, I think you tried to unmute yourself before to ask a question. and maybe you were having some troubles. Would you still like to ask a question, or?
Yes, sir. I have some questions. Am I audible to you?
Yeah, I can hear you.
Yeah. What is the, you told that the manganese manganese is mainly deposited in the shallow water environment. And okay. So what can be the pH level of that water? Means it is the acidic water, or what is the situation of the water of those basins? Means those shallow water conditions, it is acidic, or what type of things is there? pH conditions I want to know.
This was we didn't work on. It's a great question. We didn't try to model pH, but there is an interesting work from Ite Alevi, who modeled the evolution of Precambrian pH, and it's likely some sort of circumneutral pH, not acid. So somewhere between
Neutral pH. Okay.
Yeah, somewhere between 7 and 8, I think so, if I still have good memory of this article.
Okay, that means the water is not acidic.
No.
Okay, thank you, sir. Thank you.
Welcome.
Okay, we have another question that was typed in. I think you may have touched on the answer to this one when you were talking with Don. The question is, what do you think are the characteristic spatial and temporal extents of the oasis?
So what we can see in terms of the geographical extent, we saw that because we basically studied so far the lower part of the Mozan groups, but some evidences have also been shown in another sedimentary succession, which is the Suzy, where the evidence of localized surface oxygenation was also demonstrated by an article published in Nature Geoscience by a colleague here in Tuebingen, showing that at least from the Suzy to the part of the Mozan group that we've studied so far, the oxygen oasis or the localized surface ocean oxygenation was still persisting. So we have an age between 2.98 and roughly 2.85 billion years that we see persistently biogeochemical proxies for localized surface ocean oxygenation.
Okay, and that question was from Paul Mueller, and if you wanted to add any more to that question, you can go ahead and unmute yourself. But otherwise, Paul Hoffman had a question as well, and he asks, to what extent did the ambient crust and igneous activity consume O2 and delay the rise of oxygen as originally proposed by Berkner and Marshall, 1965?
Come again? Can you come again, please?
To what extent did the ambient crust and igneous activity consume O2 and delay the rise of oxygen as originally proposed by Berkner and Marshall in 65?
From the data we have here, we cannot really tell because we studied only shallow marine sedimentary successions, but it's an Archean oceanic environment with a lot of oxygen buffers, but what is happening with the crust, we don't really know, but model shown that the oxygen didn't go to high in the atmosphere, and then the level was still below the ratio that was reached at the great oxidation event during the Paleoproterozoic. It's a great question. I will think about that, but the study that we have done here doesn't allow to answer to this question, because we only show that the oxygenation occur in shallow marine water. We did some uranium isotope from some continental setting, but it's only very mildly oxygenation mobilizing uranium in the continent, but the level was still very slow. So for arguing for an oxic atmosphere during the Archean, so it was still anoxic for what we see.
Okay, Paul, if you have anything else to add, you can go ahead and unmute yourself. But otherwise, Johanna Carbone had a question as well, and says, what about,
Yeah.
Oh, it says, what about meteoric fluid involved in the formation of rhodochrosite?
Meteorite?
Meteoric fluid, sorry. What about meteoric fluid in the formation of rhodochrosite?
But I think we answered this question in early work on publishing Precambrian research in 2016, where we did first what was primary mineral phases related to early diagenesis, and what was associated to any late diagenetic processes, and then we separated what was affected by post-depositional or more modern alteration, and we only kept what was related to early diagenesis in the Mesoarchean environment, and there is no evidence of meteorite influencing the samples that I've shown today. It could have been possible, but the samples that I've shown today are not affected by any late diagenetic process. We did clay mineralogy to further check, and then we have a sort of elite phases with still a small amount of smectite, like less than 5% of smectite preserved in this elite, and the polytype of this elite is characterized by 1MD type, which is a resultant of diagenetic solid state transformation of smectite to elite during diagenesis without involving any fluid rock interaction during the precipitation of this carbonate. So they were quite well preserved. So no post-depositional alteration involved there.
Okay, Johanna, if you had anything else you wanted to bring up, you can go ahead and unmute yourself.
Yes, thanks a lot for the talk and the question. I was not thinking of a post-depositional setting. I was thinking that the rhodochrosite could be primary, but could have involved some meteoric water during this formation of early stage diagenesis at the time of deposition. And that could explain why you have only on the shallow water and not more on a deep water part.
But what I've shown, we also have rhodochrosite. I don't know if I can still go back. Also have rhodochrosite in like here. I don't know if you can see here. So this is between fair weather and storm weather wave base. So it cannot be. I will not think of meteorite water having too much influence at this depth.
Okay, thank you, Frantz. I'm not getting any more questions.
Maybe I can ask Alex.
Go right ahead.
So Frantz, about this phosphorus aluminum ratio that you used, I'm wondering, so you seems to be implying that was a high phosphorus content in seawater in deeper part of a basin, which is sort of inconsistent with what we think should be in ferruginous waters. And but on another hand, can you separate phosphorus content in seawater versus processes that remove phosphorus into sediments? Like, for example, if you have high organic carbon burial, you would incorporate more phosphorus, whereas aluminum ratio more controlled by provenance and weathering. So how do you separate between organic carbon burial versus phosphorus content in seawater?
It's a great question, Andre. It's difficult to separate, especially in shallow water, because like we saw, like we demonstrated in the 2018 EPSL paper, that most of the primary organic carbon deposited there was remineralized completely to form carbonate. So we don't have any idea of the primary organic carbon that will have been deposited initially to try to understand what is really the origin of this low phosphorus concentration in shallow marine environment. But it's just a remark that we did that I wanted to show here, that it seems to be like low phosphorus concentration preserved in sediment compared to what we see in deeper water, where we don't have carbonate being precipitated, and where the organic carbon is pretty well preserved compared to shallow water.
What would be a ratio of phosphorus to total organic carbon content if you would plot, if you would calculate it for both? What would be the ratio of phosphorus to total organic carbon content for shallow and deep water, if you calculate it? I don't know if you calculated them, but
I don't really understand you very well. Maybe.
So what if instead of phosphorus to aluminum ratio,
Yeah.
what is phosphorus to total organic carbon content?
Phosphorus to total organic carbon content will tend to be high in shallow water, but this is because the organic carbon has been remineralized by manganese and iron oxide during diagenesis. We cannot really know, because the value that we'll have now will be much higher than what was initially. So this is what I was trying to explain. You can only do it in deep water because you don't have carbonate that oxidize organic carbon during diagenesis. But in shallow water, it will be just a speculative value that you will have.
Okay, thank you.
That's why we didn't try to use this phosphorus carbon ratio initially, because we clearly shown that we have abundant carbonate, and this carbonate is like mainly derived from the organic carbon remineralization during diagenesis.
Great, thanks, Frantz. It looks like this generated a really good question session. Last call, would anybody else like to ask a question? Okay, then I think with that, thank you very much, Frantz. Everybody, thanks for being here and thanks for participating in the question session. And so next week we'll have Dr. David Corrigan, and I hope to see you all there.